Now, in an earlier lecture, talked about the fact that the cytokines will have an effect on the neighboring endothelial cells lining the nearest blood vessels, whether they be the capillary beds, the arteries, veins, whatever. And that as the white blood cells are flowing by, they will be told to slow down, stop, and to exit here. Well, those molecules that the endothelial cells will express on their surface are known as cell adhesion molecules. The cell adhesion molecules act as, road signs. Tell them stop here, slow down, come here. The different types of these recruitment molecules that tell the white blood cells to slow down and to exit here, we see they're known as selectins, P and E selectins, leukocyte functional antigens, LFAs, LFA alpha, LFA beta, and then the intercellular adhesion molecules, ICAM, ICAM1, ICAM2. Each has a different role. Each will be expressed by cells at different ratios. If you look at the selectins, P-selectin, E-selectin, overall, the selectins, their job is to bind carbohydrates on the surface of our white blood cells. They start the interaction between the flowing leukocyte and the endothelial cells lining the blood vessel. Because before this, the white blood cells and the endothelium have no interaction. They do not respond to each other. The integrins, they bind to specific adhesion molecules on the surface of the white blood cells. LFA alpha, LFA beta work together as a dimer. Depending upon which ones, alpha L, beta 2, alpha M, beta 2, there's different versions that react with different cells. Different concentrations on the endothelial cells cause different activities of what's going on here. Notice ICAM though. ICAMs, ICAM1, IC3B, that may be on the surface of, bound to the surface of, monocyte, neutrophils, macrophages, NK cells. This is a strong adhesion. This, with selectins, is a very weak adhesion. Strong adhesion, very tight. The ICAMs. These are also going to be expressed on the surface of endothelial and leukocytes. These are going to interact very strongly. LFAs on the surface of the monocyte, the neutrophil. ICAMs are mostly going to be on the surface of the endothelium. Maybe a few leukocytes. So what you have is the P-selectin on the endothelium interacting with sugars and whatnot on the monocyte macrophages, the LFA on the surface of the white blood cells interacting with the ICAMs on the surface of the endothelium. Weak association between these two LFAs and the ICAMs, a tight association. So, neutrophil here in our example, CR3, which is the alpha M beta 2, as well as LFA1, alpha L beta 2. Different subunits, slightly different protein structure. Overall fit within the LFA family. CR3, LFA1 interact with the ICAM family. Notice here ICAM1, ICAM2. They have these set conserved domains. Endothelial cells that have been activated due to cytokine chemokine will express these ICAMs on its surface. Which ones, depending upon the what the concentration of cytokines and chemokines are. And I'll explain that more in a minute. So, macrophage in the tissues comes across something it shouldn't, and it releases TNF, tumor necrosis factor. Tumor necrosis factor then interacts with other cells. Some of the cells that get activated are these endothelial cells. The endothelial cells will externalize what are referred to as Weibel-Palade bodies. Weibel and Palade are the ones that figured out these granule-like things that are P-selectin and E-selectin that have already been made and basically just bunched up, stored away as one big mass inside these endothelial cells. Tumor necrosis factor from an activated macrophage will cause these P-selectins, E-selectins to be exposed out. Adhesion. So instead of flowing along nice and easily, it starts to roll along the surface of the blood vessel. As it's rolling, it's slowing down. And as the concentration of the chemokine, specifically IL-8, as they're showing here, CXCL8, as that concentration increases, we go from just E-selectins to ICAMs being expressed on the surface. So we go from a rolling adhesion to CLXR8, IL-8 receptor on the surface, doing a tight bind to ICAM, being held in place. As IL-8 crosses the endothelial membranes, this layer crosses into the blood, binds to an IL-8 receptor, which will trigger the neutrophil to push through, diapedese, push through into the tissues. And then it will basically swim or migrate up the IL-8 concentration gradient to the site of the activated macrophage that released it. Now, we don't want to sit there, if it's a local infection, we do not want to jump over to systemic response, because systemic responses with TNF-alpha and interferons and whatnot can lead to systemic inflammation. Systemic inflammation that is not kept in check will lead to septic shock. Septic shock meaning that you will get loss of blood volume, heart has to race to keep up with it, you will go into shock, and there is always a chance of death. Because of this increase in cytokines, chemokines in response to a systemic response, you can also end up with disseminated intravascular coagulation. Basically, you start throwing clots. Clots in your arms, clots in your legs can be dealt with, but when those clots move, get into the major bloodstreams, and then go to the heart, go to the brain, then you impede blood flow and you get heart damage, brain damage, and resulting in death. So, by keeping things local, you get a local inflammatory response, you will get localized clotting. That'll slow down the flow of blood, increase the inflammatory response in the area, and then you will see that later those clots will be removed and that excess fluid will also be removed. Problem if it's disseminated, if it goes systemic, you get multiple blood clots through multiple different capillaries, through multiple arteries and veins. These move, these get caught up, form bigger and bigger clots, and like I said, you either get inhibition of blood flow to the heart or to the brain, resulting in death.